Okay, I think we'll get rolling, folks. Okay. We are beginning the so-called clinical part of the course, where we're going to focus on individual species, genera, strains, what have you, that cause particular human diseases. And to start with, we're going to deal with the Enterobacteriaceae, Vibrio, Campylobacter, and Helicobacter. Okay. As always in my lectures, well, not always, but usually the colours mean something. We're going to, in this case here, we're going to have some general definitions that will apply to a lot of stuff. But basically, pretty well up to here, we're going to be talking about the Enterobacteriaceae. Vibrio cholerae is an organism, is not a member of the Enterobacteriaceae, and nor is Campylobacter, and nor is Helicobacter, which is why they're in red. Okay, so we're going to cover four different groups, quite different groups of organisms today. The major focus being on the Enterobacteriaceae. Now, the reason we're focusing so much on the Enterobacteriaceae is because they're one of the most common causes of opportunistic infections in hospitals. And there's a huge number of diseases, and there are actually a large number of species that cause human disease. And I don't propose to sort of have you memorize about 60 different species. I've just mentioned a few of them here in passing. I may mention another one or two, but we're going to mainly be focusing on Escherichia as a genus, as a species, and we're going to talk a lot about Escherichia. But basically, there's a huge number of these different species that cause opportunistic infections, and the key word is opportunistic. They basically cause disease when they have an opportunity in this case being particularly in hospitals. Now, we're not just covering situations in hospitals. Some of these organisms don't cause opportunistic infections as such. Although any organism that could cause a disease in a healthy person obviously could cause it in a non-healthy person, because it's a lot easier to cause disease in a non-healthy person, of course, than it is a healthy person. But some of these organisms cause disease in the community or individuals. And so we're going to cover specifically some of the members of the Enterobacteriaceae, some instances strains within a species that have particular virulence factors. In other instances, we're going to cover particular species or genera within a species that cause particular diseases, and I'll explain what I mean when I get there. Okay, so first of all, we're dealing with these opportunistics, which are particularly, again, the Enterobacteriaceae and these huge number of species that we're talking about. And you'll become associated with the names as the years go by. In private practice, you may not be so familiar because these things will not be cultured probably, but in hospitals, they're going to be cultured and characterized, so you're going to become comfortable, some of you anyway, with a lot of the organisms by experience as the years go by. Okay, so the Enterobacteriaceae. Now, I've mentioned already opportunistic infections, okay, as an overview, what goes on in hospitals. And the other thing I said we're going to be covering among the Enterobacteriaceae are specific, particularly gastrointestinal diseases that are caused by this group, but we'll mention a few other things in a little bit. Escherichia coli, many of you have come across Escherichia coli as being something boring and dull, the organism that everybody works with if they work with gram-negative bacteria, or bacteria period. But there are strains of E. coli that are major causes of gastrointestinal disease, and that's pretty obvious with what's been going on in the newspapers the last few days with the enterohemorrhagic E. coli, but clearly not all E. coli are boring, dull things that you just work with and play games with in high school or something with. Some of these organisms are major causes of gastrointestinal or invasive disease. Among this, the Salmonella are a major cause in the community of gastrointestinal diseases, as are Shigella. Yersinia enterocolitica is nothing like as common, but it's particularly common in Scandinavia and has been recognized more in this country as the years go by. So we're covering, again, the organisms that cause diseases in healthy, or for that matter, sick people, but there's organisms that are particularly thought of as causing outbreaks, small epidemics, and particularly affecting healthy people. I'm going to mention Reiter's syndrome here, which is a disease many of you may not ever hear of again, but I find this disease still very fascinating. I used to work on it a while ago. The Reiter's syndrome is associated with the histocompatibility antigen HLA-B27, and it's a form of reactive arthritis, rather like another disease we're going to talk about later, rheumatic fever or rheumatic arthritis, as opposed to rheumatoid arthritis, which we'll talk about in the Streptococcus lecture. Reiter's syndrome is a disease which is set off by an infection, often of the gastrointestinal tract. It can be the urogenital tract, particularly caused by Salmonella, Shigella, or Yersinia. There are also non-Enterobacteriaceae, including Campylobacter and Chlamydia, which have been shown to initiate Reiter's syndrome. The local site is an infection, but the organism does not spread and does not almost never spreads, and it is not an infection of joints. It is an inflammatory disease resulting from an infection at a focused site such as the gastrointestinal tract or the urogenital tract. And that's Reiter's syndrome, associated with ankylosing spondylitis, which has a much higher, this has about a 75% association with HLA-B27, whereas ankylosing spondylitis has more like over a 90% association. And I don't want to say any more about it than that. It is an interesting disease, and I do remember about 25 years ago when I was at Southwestern Medical School in the rheumatology department there. One occasion there was a patient that showed up, and I happened to be a person who was walking between the infectious diseases and rheumatology department, and what my interest. And the rheumatology department actually got together with infectious diseases to deal with this patient because it was such an unusual, strange thing. But anyway, so you may never see a case of Reiter's syndrome. Who knows? But it's something, at least you've been exposed to it. Okay, but of more general importance, Enterobacteriaceae. I've mentioned again that these are particularly, we think about the Enterobacteriaceae as being one of the major causes of diseases in the hospital, along with diseases such as streptococci and staphylococci. And, it tends to sort of vary from decade to decade and hospital to hospital, what the particular problem is in a particular place. But in general, Enterobacteriaceae are one of the major groups of organisms that cause hospital-acquired infection. As regards in community-acquired infection or otherwise healthy people, a few things stand out. For example, Klebsiella pneumoniae is commonly associated with respiratory diseases. It has a particularly prominent capsule, and that's a common theme that we've discussed in the bacterial pathogenesis lecture. If you've got a particularly prominent capsule, it can often be involved in pathogenesis, and this appears to be the case here. As regards urinary tract infections, the most common one is E. coli, and it is believed that what happens is that people when they're sort of wiping when they've been to the bathroom or whatever, it can spread from the gastrointestinal tract to the urinary tract. So this is a rising infection that rises from the outside going up the urinary tract inside, as opposed to what we're going to be talking about with the enterohemorrhagic E. coli, where the effect on the kidneys and on the urinary tract is from the inside going out. Okay, so this is the source, fecal contamination, usually in this case. The only reason that I mentioned Proteus here, the vast bulk of the cases most urinary tract infections are gram-negative in nature, and many of them are caused by E. coli. The only reason we mention Proteus here is because the National Board loves questions about weird things. And Proteus happens to be an organism that has a prominent urease, and it degrades urea. And so the urine is often alkaline in a Proteus infection. Okay? So it's not that I consider it particularly such a big deal, but it's something that's why I'm mentioning it. Okay, so often if you get an alkaline urine, you know you have a Proteus infection. Okay, again, still with the Enterobacteriaceae, which will be most of the stage. But I've got 2 lectures on this, I should have pointed out, and there's about 66 slides I calculate. So we're going to do half of it today. We're going to stop halfway through the Salmonella, and the next lecture will finish the rest of it. Okay? Okay, the Enterobacteriaceae are gram-negative facultative anaerobic rods. And I don't have to explain that again because you all know very well from the basic part of the course what a facultative anaerobic rod is. We didn't cover this. These organisms are oxidase negative. That is, so there's no cytochrome oxidase, which is part of oxidative phosphorylation. And the reason that's brought up is because it's one way you can often differentiate between the Enterobacteriaceae and things like the Vibrios, or the things that are not members of the Enterobacteriaceae. And so you'll see the opposite statement when we get to, I think it's Vibrios. So oxidase negative, they produce an enzyme which you can easily test for. It's the simple change in colour of a dye. And it tells you whether this enzyme, cytochrome oxidase, is present or not, which is often characteristic of the Enterobacteriaceae. Okay, we're talking a lot about feces today. And those of you who didn't catch it in the news can guess what it has to do with spinach, for example. We'll get more into that as we go along. Feces, that's what we're thinking about a lot today. Not a pleasant thought, but regardless, that's what we're thinking about today. Okay, E. coli are lactose positive. They ferment lactose. They produce a positive reaction in a test for lactose. You don't usually, and I'll explain in what situations I'm talking about shortly, you don't usually you don't usually identify the E. coli by this test. They are lactose positive. Because many of the organisms that we find that are normally found in the gastrointestinal tract are lactose positive. And so without doing a lot of work, because the feces contains an awful lot of different bacterial species, without doing a lot of work, it's very hard to straighten out whether you've got a lactose positive E. coli from something else. However, you don't usually have Shigella, Salmonella, or Yersinia in the gastrointestinal tract, because these are organisms the reservoir is a human being. And that's a term you'll hear a fair bit in this course, the word reservoir. The reservoir for these infections is actually other people. And so you don't usually see a lot of lactose negative organisms in a fecal, when you culture a fecal sample. And so if you do find a lactose negative organism, it doesn't mean it's automatically Shigella, Salmonella, or Yersinia, but it means you've got a good shot that it probably is. So the focus here on fecal samples is more on Shigella, Salmonella, and Yersinia rather than E. coli. Now, the situation with the feces is totally different to anywhere else. You should not have bacteria in your bloodstream. If you have bacteria in your bloodstream, you're sick. Simple as that. There's something wrong with you. You shouldn't have bacteria in your bloodstream. You should have bacteria anywhere else inside the internal milieu proper. And if you've got a gram-negative organism in particular in your bloodstream, which is growing and multiplying, it has a good possibility that it's going to produce an endotoxemia. It's going to produce a toxic shock, the classical toxic shock, endotoxemia. In addition, many of these gram-negative organisms, and we're talking about some of them today, have particular toxins. If they're present in the gut, they may or may not do you any harm. But if they're systemically floating around in your body, they're certainly going to do you some harm. And so any time you've got an organism that's systemic, or I won't say any, nothing is absolute in this world, but almost always you're going to culture these things if you suspect that you've got a systemic infection and you want to really know what's going on. Okay? And it's pretty obvious usually, that not always again, but it's pretty obvious because the person's listless, they're weak. You've all had, viral or bacterial infections, you know what it feels like. They're listless, they're weak, they're drowsy, they don't feel great. They have stomach problems in this particular case. So if somebody's got in the bloodstream, you're going to culture. And so what I said about E. coli certainly doesn't apply if you isolate E. coli from somebody's bloodstream. You're going to want to know what it is. You're going to do biochemical tests. And I showed you a picture in the earlier slides taken at the VA about 20 years ago, and things haven't changed much. But basically, you're going to do simple biochemical tests. There are simple, sometimes it's done with a machine, sometimes it's done manually. But the bottom line is there's a series of tests that are done, and it just spits out an answer. And if it's half a dozen come up positive, it'll be that organism. If another half a dozen come up, it'll be that organism. And that's what's done generally to particularly sort out the Enterobacteriaceae, because there's so many of them, and they're not always going to have the same antibiotic that they respond to. So it's useful to have, most of the time you'll be okay, but now and again you get an organism that just happens to be resistant to a particular antibiotic, and it's useful to know what the species is for that purpose. And people who have experience in the clinical microbiology laboratory or infectious disease physicians can oftentimes know that if they see something, a particular species there, we'll get this right in a minute, they'll know that they may choose a particular antibiotic. And that's something that's generally not taught at this level. That's just something that's done more by experience as you work with this stuff over the years. Serotyping. We've mentioned the O, A, and K, the OH and K antigens in passing before, and I now want to emphasize them again, particularly because OH and K typing is very important when it comes to the Enterobacteriaceae. It's not something that you really think about in many instances. First of all, they don't even have gram-positives. We know already wouldn't even have an O antigen, so we wouldn't be talking about gram-positives altogether. But in many cases, we don't care about these things very much. But we do care about them for a variety of reasons that will become clearer as we go along. The O antigens, I remind you, are the outside of the lipopolysaccharide, the variable portions of the very variable portions of the lipopolysaccharide that can differ and do differ among species. The H antigens are the flagellar antigens, and the K are the capsular. And that's why we talk about O157H7. You're talking about the O antigen, the lipopolysaccharide, or the H antigen, the flagellar antigens. And that's just one case where it's used. It's used a lot in epidemiology, particularly with salmonella, and we'll see why shortly. Diarrhea and dysentery. I just want to define those terms. There's often an overlap. We can see situations where you see basically almost exclusively a diarrhea. There can be situations where you see almost exclusively a dysentery. There can be sometimes you see both, and sometimes it particularly correlates with a particular organism. Nothing's perfect in this world. Nothing is absolute. A diarrhea, I think you're all aware what diarrhea is. You may not be aware what a dysentery is. A diarrhea is where you have a copious secretion of fluids into the intestinal tract, and the feces becomes very fluid and sort of, wet and gooey and what have you. And it's very unpleasant, as we all know. Dysentery, on the other hand, is referring to what happens to the lining of the intestinal tract, and it refers to the fact that there's going to be injury going on, and there's going to be infiltration of white blood cells often, which may show up in the feces, or there may be damage to blood vessels and tissue, which means there's going to be blood's going to show up in the feces. So that's a dysentery as opposed to a diarrhea. And I do not mean these are absolute things that never happen together, as I said already. But they are terms that we use, and particularly, for example, with Shigella, we refer to it as bacillary dysentery. And when we talk about cholera, it's a very potent, very nasty form of diarrhea, which if not treated, will kill you from dehydration. So there are particular cases where we see diarrhea more associated, and the cases where we see dysentery more associated. But you can have a diarrhea and a dysentery, some instances going along together. It's not an absolute thing. What I've tried to do as we go through, some years ago, Richard Hunt discovered, before he actually was charging for it, discovered Dennis Kunkel's website. Now, actually, it's free to this medical school. I don't know why he doesn't charge us. But anyway, he has some beautiful pictures there. And they're electron micrographs. It doesn't matter a bean. What I've done is they would look like this basically look like this under a gram stain, but not as much detail. And what I've done to go through this lectures, I show you pictures of different bacteria, and they're either labeled, they're pink or they're red, to try and emphasize whether they're gram-negatives or gram-positives. Sometimes we'll see some weird shapes or things that are different. For example, you'll notice here that these rods are straight. I suppose you'll see a picture of the Vibrios later on where they are curved. You'll see the Campylobacter where they're pleomorphic. Okay, so some of this is going to be quite repetitious, but you'll get the idea as we go along. Okay, so these are coloured. They wouldn't look this way, of course, under electron microscope, but they've been coloured to emphasise a point. I think the Mycobacteria might have coloured yellow when we get there. I don't remember. Okay, so this is what a typical member of the Enterobacteriaceae looks like. Escherichia coli. There are actually, the taxonomy continuously moves on and at one point I was interested in taxonomy and I sort of liked it. But as the years have gone by, I've begun to understand the frustrations of clinical microbiologists and physicians. The issue really comes down to a particular group of organisms is associated with a particular disease. That's what we care about. That's what means something. Scientifically, it may be totally incorrect, but that doesn't really change reality. And so, anyway, in this particular case, there's more complexity of the Escherichia coli than I've got into here. But I'm going to just live with Escherichia coli for now. For example, Escherichia coli and Shigella, by any scientific basis, would be considered the same species. We never call Shigella E. coli or vice versa, because over the millennia, or not some millennia, over the decades, we've basically always called a Shigella, and we've always called E. coli an E. coli, and most people continue to do this regardless of anything else. Okay, so that's what we're going to do here. They're genetically very similar. In fact, more than similar, they're actually same members of. They're just different species in the same genus. And not surprisingly, they're so closely related genetically, it should be no shock there are many similarities in their diseases. And you'll hear me talk about one instance with the E. coli, you'll say, well, it's Shigella-like. In another case, you may hear me say it's Salmonella-like. And that's, really not a surprise. Okay, the enteropathogenic E. coli. There are five actually, but one of them I've not got into because I think it's so rare. But there are a number of different groups of E. coli which have very specific characteristics. That doesn't mean you'll ever know this if you see a patient with it, because usually you wouldn't ever go ahead and go that far to see what's going on. But we do know what's going on. In the case of the enteropathogenic E. coli, these are organisms that are actually not penetrating. They're actually sticking to the surface of the epithelium, and they're doing damage at this surface. And particularly you see a fever, more diarrhea, vomiting, nausea, and a non-bloody stools because it's not damaging this epithelium a great deal. Okay, so that's enteropathogenic, and you'll see that it's there, not there or there, and you'll see that there's no toxin mentioned. But it's not emphasized in this particular group of E. coli. Now, the word says exactly what it means. Enterotoxigenic E. coli. Doesn't mean it's the only E. coli that produce toxins. There are a particular group of organisms that have particular characteristic. This is a diarrhea that resembles cholera, Vibrio cholerae, which is not a member of the Enterobacteriaceae, but there's a toxin that's similar, and it's not that difficult to pass things among these organisms, even if they're not members of the same family. And so these have a diarrhea like cholera. It's milder, and it often occurs when people are going to places like Mexico, so it's referred to as traveler's diarrhea. It's not the only form of traveler's diarrhea, but it's one form of it. That's enterotoxigenic. Now, when you're talking about the enterotoxigenic, as opposed to the enterohemorrhagic E. coli, here we have two particular toxins, heat labile toxin and the more recently discovered heat stable toxin. The choleragen refers to cholera toxin, which is the major toxin and which was discovered many years before the E. coli toxin. And that's why it's referred to as choleragen. It's not referred to as E. coligen or something, because cholera was the disease that was discovered first. And this toxin does things like cholera toxin. An adenyl cyclase is activated in the surface of epithelial cells, resulting in the production of cyclic AMP, which in turn leads to secretion of water and ions into the intestine. That means that the person becomes dehydrated. Now, in the case of this disease, I mentioned already, it's mild. So, the chances are the person is not going to die from this. Now, in the case of cholera, it isn't mild, and the person will die from it usually if not treated. The heat stable toxin, on the other hand, which is exactly what it says, I don't know what temperature, it's not that important, but it's more stable to heat than the heat labile toxin. That's how they're differentiated. It produces uses to act the production of guanylate cyclase. In this case, the amount of cyclic GMP goes up, and it has the same effect, but this causes the intake of water and ions into epithelial cells, to go down. So in either case, they're different mechanisms, but the same thing's going to happen, dehydration. And that's the big deal in an enterotoxigenic E. coli infection. Again, the organism is non-invasive, enteropathogenic, enterotoxigenic, and a couple of other groups I'm going to talk about, one more group I'm going to talk about, they don't invade. They don't get into the bloodstream. They stay put in the intestine. And again, the word... One second, I'll answer first. The word invade can mean two different things. Invade can mean invade the epithelium. Invade could mean getting into the bloodstream. You have to listen to what I'm saying, read the notes, and to what I'm talking about. The word invasion is a very vague term. Okay? Go ahead. I think Dr. Ghaffar has gone further than I've gone and he's got a slide that shows the different proteins involved in the process and the actual mechanism. I've chosen not to go that far with it. Okay? But there are some receptors in the cell surface and other proteins that are regulator proteins, and they will actually be hit by this toxin, and these regulator proteins will be up or down in activating these receptors and resulting in production. There are enzymes there that will result in production of cyclic AMP as part of the process. Okay, but I'm not going to go into that sort of detail. Okay, I don't think you need that sort of detail. Okay, but that's what's going on. Okay, so talk to Dr. Ghaffar. He knows more about this than I do. If you want to know more. Okay. All right. Any other question? All right. Enteroinvasive. The enteroinvasive E. coli are quite distinct from the enteropathogenic and the enterotoxigenic E. coli. The enteroinvasive E. coli are, again, that's exactly what I said, but I didn't say invade into the bloodstream. I've specifically shown you this cartoon here. So we're seeing the cell has invaded into the epithelium. And I don't want to see any confusion in this course. Now, if I don't emphasize whether something invades an epithelium or invades the bloodstream, it doesn't mean it's unimportant, but I haven't mentioned it, so you're not responsible. But if I do mention it, then I've probably got a reason, good or otherwise, to do it. And so remember, the key issue is not the word invasive, but the issue is invading here of the epithelium, not getting into the lumen and not getting into the bloodstream. This is not a systemic disease. And we care about this a lot because you treat somebody. A lot of times, if somebody's got a gastrointestinal infection and they don't feel, and they may feel a bit bad. They may take a day or two off work. In many cases, they don't even go see a physician. And you can't buy antibiotics over the shelf. So many times, people don't even get treated if they have a gastrointestinal infection. The only time you're going to see somebody is if they're really in bad shape. And in some instances, it's standard protocol that you don't treat with an antibiotic because it doesn't seem to do much except cause the organism to be shed more and cause infections in other people. So when we're talking about a gastrointestinal infection, the big deal is how bad is it? When something gets into the bloodstream, as I've said already, there's no discussion how bad is it. It's bad. Okay? So that's one of the reasons I'm making this big deal about invasiveness, not because I particularly care whether unfortunately for the board you do have to care, but not because I particularly care whether it invades an epithelium, because it makes a big difference if it's in the bloodstream or if it's local. Okay, now we are at the new spinach disease. Okay, enterohemorrhagic E. coli. And this is why we care. You've all been watching the news. The newspapers. There's been about 100 people all over the country now have died from it. I think there's only one or most two people have died from this so far. But there's been 100 people who've got sick. This is an epidemic. And you all know that it's resulted in spinach being withdrawn from the shelves all over the country. I mean, we aren't withdrawing them from the shelves for enteropathogenic E. coli. We aren't withdrawing them for enterotoxigenic E. coli. We're withdrawing them specifically for this disease. So clearly it makes a difference what disease we're talking about. And I do use that word in the real world because, the classroom or sometimes the research lab is not the real world. Okay? Now, I don't know, the investigation's not been completed, but presumably what we're talking about here is that somebody's been treating these on some farm somewhere, has somehow let manure, possibly from cattle, some other animal, who knows, but particularly cattle, has got on the spinach. I'm assuming this is, and I don't know this, and I'm not sure anybody knows it, but we certainly would think it was not from people, because if it was from people, it would mean that those people were already infected with enterohemorrhagic E. coli, and presumably some of them would have the disease. So, anyway, that's the sort of thing that goes on. This was first described, it wasn't that long ago, I don't remember, maybe certainly since I've been teaching medical students, maybe 15, 20 years ago, I don't know. It was originally described as an infection at the Jack-in-the-Box in Texas, where a group of people got infected by eating hamburgers, and that's why it does have, and microbiology does have an effect, just like going on a plane has an effect in whether you can take water on. Well, we all know that it's a requirement now when you cook hamburger that it has to have a certain temperature inside the meat, otherwise, it's not acceptable to eat. And any hamburger joint in the country now will be doing that, or they're going to have lawsuits. So that's where we started with enterohemorrhagic E. coli, and it's traditionally been thought about as being associated with hamburgers. But it can obviously be, it's been known that it does occur in contamination of vegetables, and you see another example today. And we'll probably forget about spinach after a couple of years. It will be something else. Who knows? Maybe next time it will be tomatoes. I don't know. But at the moment, that you've had an example of how you can get this infection, but it's been particularly associated with hamburgers in the past, or uncooked meat, or poorly cooked meat, I should say. By the very nature of the fact it's H7, that immediately tells you, we're talking, not all these organisms necessarily are flagellated in the Enterobacteriaceae, but this one's got a very, very prominent form of flagella. You can see how huge these things are. And the O antigen again would be 157. Okay, so we particularly talk about the OH and sometimes the K antigens. In this case, the K antigen is not particularly associated particularly with this group of organisms. It may be variable, but what is the big deal in this case is the O and the H antigens. And I don't emphasize it anywhere else because there's no reason to emphasize it anywhere else. But any member of the Enterobacteriaceae could have an O, a K, an O and H, and if it has a capsule, a K antigen. And I said, this slide was put together before the last couple of weeks, so it doesn't say spinach. And that's what I keep emphasizing. Microorganisms are living things, and they change from year to year. And this one's changed in the last couple of weeks. So we have to be ready. We have to learn certain things for exams. But once we get into the real world, we have to realize every year we have a different group of organisms that becomes important for 1 year or 2 years, and then in many cases rapidly nobody cares about it anymore. But anyway, this disease, particularly with meat, hemorrhagic is the bloody, copious diarrhea, which there are fused leukocytes, and it doesn't usually produce a fever. When it's referred to as hemolytic uremic syndrome, which is the nastier form of it, you see a hemolytic anemia, a thrombocytopenia, and kidney failure, and that's why this disease is such a problem, because the organism gets into the bloodstream and again descends from the bloodstream through the kidney and can cause kidney failure. Go ahead.
Should it be considered dysentery diarrhea?
Again, don't ask me why this happens. These are, particular diseases, they get defined this way. I mean, bacillary dysentery, for example, is called bacillary dysentery. You do see diarrhea in bacillary dysentery. It's just common usage, So that's the most common thing that you see. I'm not going to say dysentery could never happen, okay? I mean, clearly it could because it produces a potent toxin, which you'll hear about in a second. Okay. Enterohemorrhagic E. coli, verotoxin, referred to it, and I noticed in this year's Murray it referred to it as sugar rather than sugar-like. And so the word like, when you see it on the end of something, what it usually means is that another disease was described first. And then the secondary disease came along, and so it gets a name where the word like is attached to it. Now, I'm going to try and stick with these words like, but that doesn't mean somewhere else outside the medical school. I have no control what goes on in the whole United States. Somebody else might use a different term, and they're probably correct. Because as the years go by, people forget that oftentimes that something was like another disease, and so they leave the word like off. Okay? So it might be referred to as, it might not say the word, it might just say sugar toxin. But anyway, this verotoxin, as it was originally called, is like the sugar toxin of Shigella, which is a major pathogenic disease that we've known about for many, many years. There is also, not surprisingly, you mentioned the problems with what happens in the blood. It produces a potent hemolysin as well. Okay, so there's two toxins associated with enterohemorrhagic E. coli. And that doesn't mean that they're the only E. coli that can ever, under any circumstances, produce virotoxin, because an E. coli is an E. coli is an E. coli, and things can get transferred on plasmids and phages and what have you around between one organism and another. So it's not an exclusive thing, but particularly the problem is that this organism, say, gets into the bloodstream. And if you're producing a virotoxin in the intestine, it may be one issue. It may be an issue if you're producing hemolysins in the intestine, that may be one issue. But if you're producing these things and secreting them in the bloodstream or in the kidneys, that's another issue altogether. And that's why these are such a big deal. We talk about enterohemorrhagic E. coli, not just they're the only ones that produce it. The treatment for gastrointestinal diseases, it might not be what you would expect in first principles, if you're just thinking using your common sense. The first thing is not antibiotics is what you think about, it's fluid replacement. Indeed, why cholera is not the problem that it used to be, which is the first disease that you think about when you talk about fluid replacement, because it's such a massive water secretion in cholera, is that it's the dehydration that kills you. A lot of times if an organism is in the intestinal tract, it may make you uncomfortable, it may make you have a diarrhea or a mild dysentery, but eventually it will go away. Okay, so you oftentimes, again, the issue is how severe is it, and how do you clinically judge whether you're going to do your patient any good? It's not a textbook issue, it's a real world issue of how good do you think you're going to do your patient and what happens in a particular situation. And as again I mentioned, if you've got something in the bloodstream, you should be pretty well know by now what you need to do. Okay, so let's talk a little bit about pathogenesis factors now. There are the fimbriae, and another word for fimbriae is the pili. The two terms are often used interchangeably, and I shall use them interchangeably here. Pili or fimbriae. These are these little appendages that I've shown you a picture of before that you find on many gram-negative and gram-positive bacteria. This is an example of where, this is a slide we showed in the pathogenesis lecture, and I said that when we get to particular lectures we're going to bring these things up again and go over them a little more if we have the time. In this case there are many different types of fimbriae, but two that are particularly well characterized are the type 1 and the P fimbriae. Different strains of E. coli can have type 1, which binds to mannose, it binds to glycoproteins on epithelial cells, or it can bind to galactose components of the cell surface, glycoproteins and lipids. And they're not particularly associated with a particular disease. They've been studied a great deal in urogenital infections, but they're there and they do presumably contribute, but this is one thing among many. So those are that's E. coli. Now there are other species of E. coli nowadays, but they're very rarely involved with human disease. And when we mostly think about Escherichia, we think about Escherichia coli. Okay, Shigella. Now, things are different here in terms of what you need to know. There are 4 different species of Shigella. Any one of these species can cause bacillary dysentery. And as a good question that was asked me just now about when we see diarrhea and dysentery, I'm not saying you never see diarrhea in bacillary dysentery, but the predominant thing is dysentery, and it's been named because of the occurrence of dysentery in this particular disease. It's the classic bacillary dysentery. Another term for it is shigellosis, because in the clinical laboratory you don't generally differentiate which one of these species it is. It could be any one of them. As we said already, this is a dysentery, so there's often a bloody feces, intestinal pain, and pus, the hallmarks of dysentery. But again, not always. Now, one of the reasons, let's see if I've got it on my next slide. Well, we'll get there in a minute. There are different things that happen in shigellosis versus salmonellosis clinically, and we'll get there in a second. Okay, so shigellosis, again, these little cartoons, this is the gut lumen. In this case, it has got into the epithelium. It has not got into the bloodstream proper or into the lymphatics. There's epithelial cell damage within 2 to 3 days. Now, this can go on for quite a while. They can this organism can reside there for quite a long time, weeks. Okay, so this can reside there for quite a long time. but the epithelial damage occurs quite quickly within a few days. This is another off the website, not as nice as the other one. I think because we tried to colour this one, it wasn't coloured and didn't do such a good job. But anyway, gram-negative organisms, the Shigella, just like the Salmonella. Okay, sugar toxin. We talked about sugar-like toxin. I didn't say how sugar-like toxin worked. The other name was virotoxin. The reason I didn't do that is because it works the same way that sugar toxin does, which is why it's called sugar-like toxin. So when you see virotoxin, you see sugar-like toxin associated with E. coli, with enterohemorrhagic E. coli, you're going to be thinking about the mechanism you see in sugar toxin, which is enterotoxic, cytotoxic, and its mode of action is it actually lyses a particular bond in the 28S ribosomal RNA, stopping protein synthesis, and that kills cells. And that's why it's cytotoxic, and that's why it's enterotoxic. I haven't done this with your particular group. It's just reminded me of something. If you ever see a term like entero or cardiotoxic or neurotoxic or anything like that, you have to be talking about an exotoxin. You're not talking about an endotoxin. Because endotoxins have widespread activity on large parts of the body, on lots of different types of cells. They're not particularly fond of the heart or the brain or anywhere else. When you're talking about a toxin, it has a particular modes of action, such as in the gastrointestinal tract or elsewhere. So this would be an enterotoxin because it acts particularly onto the gastrointestinal tract. That means that maybe you don't have to learn, is that an exotoxin or endotoxin? You should know immediately, you see it, that's got to be an exotoxin. Can't be anything else. Okay? All right, shigellosis. Unlike we just talked about with E. coli, Shigella, man is the only reservoir. It is particularly common in children. And the reason it is common in children is because children love to play together. They don't always wash their hands, particularly when they go in the bathroom. They touch each other. They play with each other. And that's why it's a particular disease that's common in children. Now, that doesn't mean only children can get this disease. Some of you, I'm sure, have got young children. Mine have grown up now, so it's been many years in the distant past. But you've had situations where your kids, as little reservoirs of infection, come home and you're great, you're fine, they come down with it, and the next day you come down with it. And that's what that's what we mean by children to adult transmission. And those of you who've had it happen, and many of you have had it happen, and many of you haven't had it happen yet, will have it happen, you'll know what I mean. Now, it can also occur in adults through, and this is regulated by places like DHEC, but adult food handlers can be carriers of this disease. And if they don't wash their hands, the same thing happens. So it's particularly common in children, but it can also occur and does occur in adults. Okay, we've only got 5 more slides to go, so we've got 5 minutes, we're doing okay. Right, treating shigellosis. Again, like any disease of this type, the major issue is managing dehydration. So here's an example. Even though it's referred to as a dysentery, you can certainly get some dehydration in bacillary dysentery, even though it's not called bacillary diarrhea. Okay? So I'm sorry if that was a bit complicated. I can't give you these exact definitions that you want here because it doesn't work that way. Okay, so here's an example of managing dehydration. Now, in this case, the patients do respond to antibiotics. And the key issue is the disease duration diminished? Didn't say, does it cure it? Because it will go away on its own usually. So you might treat in shigellosis, might be in the key word, depending on how severe it is. In salmonellosis, some particular forms of salmonellosis that we will start to cover a little bit and get much more into the next lecture, there's a different situation there. Because in this case, shigellosis, it has an effect. In salmonellosis, as opposed to shigellosis, we will talk about later, they don't usually respond to antibiotics. And it just takes, you have to just wait for the course, just to wait for it to go away. Unless, as in the case of certain forms of salmonella-mediated disease that we will discuss later, it gets into the bloodstream. And I know if this sounds sort of nauseous and repetitive, it's meant to be, because I want to hit this home. Okay, these are the sorts of things we're looking at here. Okay, and again, nothing magical here. Gram-negative rods again. We all know what a typical gram-negative rod, the Enterobacteriaceae, looks like by now. Now we're at the salmonella. The saturation with the Salmonella is totally different than it is with the Shigella. Totally different. Some reason, well, there is a good reason. In the case of Salmonella, what the situation is that these are particularly transmitted from foodstuffs, from animals, from birds. It could be chickens, meat, other sorts of meat, eggs. It's particularly the thing that's transmitted from animals. And if you suspect there's an epidemic going on, as we've seen here with this E. coli situation, you want to know whether there's an epidemic going on. And the way you tell if you've got an epidemic is you have to know there's a particular strain of an organism floating around that has particular O, H, and K antigens. And so that's why people have spent their time. It isn't that some microbiologist has thought, hey, I love to play with antibodies. Why don't I see if I can find out there's 2,000 or 2003 different types of Salmonella out there? There are. And at one point in time, every one of these had a species name. We should just forget the rest of the Med Micro course, I think, at this point, and we shall sit down and learn these 2,000 names, right? No, of course not. So anyway, what's happened is, as the years have gone by, people have sort of tried to deal with this, and finally they said, well, let's just break them into three groups based on what we see clinically. We'll call one group S. enterica, we'll call one S. enteritidis, and we'll call one S. typhi. We'll call one S. enteritidis, one S. cholerae suis, and one S. typhi. Most of these 2,000 odd species, are actually members of S. enteritidis. So the general name that is out there for all Salmonella is Salmonella enterica. But basically nobody, except a few folks that are really interested in this stuff, use that term. And sometimes I've noticed recently what people are doing is they've actually gone even further, and they've actually referred to it, they may put a capital E there, or they may put a capital E there, which is totally against the rules of bacterial nomenclature. And they do it because they can't think of any other way to lump all these different things together. I'm not going to go that far, but if you ever see a situation where you see outside this course where there's a capital letter there, you'll have an idea what we're dealing with. But the S. enteritidis has got many of these thousands of different serotypes, which by some people's definition are species. S. cholerae suis is pulled out because it's got a particular disease characteristic that we're going to talk about. And S. typhi is one of the classical agents of plague. Okay? And so we want to know something's plague. We don't care that it's called S. typhi because it's one of those 2,000 names. We care because it's one of the classical agents of plague that wiped out tens of thousands of people in this planet during the Middle Ages and still does in some parts of the world. Okay, so salmonellosis. Now this is most of the 2,000 odd species, strains, whatever you want to call them, that we're talking about. Salmonellosis, as opposed to shigellosis. Shigellosis being where there's a human carrier, 4 species. Salmonellosis, where we're talking about these thousands of different names have all been lumped into one. Salmonella enteritidis, as opposed to Salmonella cholerae suis and Salmonella typhi that we'll talk about next time. Okay, this is the common Salmonella infection. I didn't say it's the most life-threatening. I didn't say it's the most deadly. I said it's the most common because it's transmitted from things like poultry, eggs, particularly from chickens, but it can come from other animals as well. There's no human reservoir. We don't generally see salmonellosis transmitted from person to person. I didn't say we never see salmonella transmitted from person to person, because we do in the case of Salmonella typhi. But I'm not talking about typhi, and I'm not talking about cholerae suis at the moment. I'm talking about salmonellosis, everything but those couple of species. So there is no human reservoir in salmonellosis. There certainly is a human reservoir in typhoid. It's referred to as a gastroenteritis, where there's nausea, vomiting, and non-bloody stools. And the thing is, it's usually self-limiting. Some cases of salmonella, certainly not. They can sometimes go on for weeks. So that's why it says the word is usually self-limiting. And that's why you generally don't give an antibiotic, because it doesn't seem to do very much as it's in terms of stopping the disease, of affecting the course of the disease. It's a thing that physicians have learned from treating patients, not something that they've got out of a book. Okay, salmonellosis. In this instance, the organism, as opposed to Shigella, which usually is on the surface here. Shigella also, as opposed to Salmonella typhi, which actually invades. Salmonellosis, the organism stays put. That's an important distinction between typhi that we're going to discuss next time. And again, in uncomplicated cases, the vast majority, antibiotic therapy is not useful. And there's no major mechanistic or huge, biological, biochemical basis for this. It's what people have found out by working with patients over the years that this is the case. I think I have one more slide. Okay. Now, I've talked about salmonellosis so far because it's the most common. We pull out Salmonella cholerae suis, which is one of the millions of names that we've talked about already, because it does something a little bit different. In this instance, it's much less common, but it's certainly not the incidence that's the big deal. But it can occasionally cause a septicemia, and we've emphasized already the issue with something getting into the bloodstream. And again, if something gets into the bloodstream, antibiotics are going to be essential. Okay, I'm going to leave it there, and we're going to talk about the causative agent of plague, typhoid, next time. We've dealt with two of the groups now of Salmonella. We'll be going on to the third group next time. Okay, thank you very much.